Now I would like to introduce speaker Dr. Ben Ho Park. Dr. Ho Park is a professor of medicine at Vanderbilt University Medical Center. He's the director of precision oncology at Vanderbilt-Ingram Cancer Center. He's also the co-leader of breast cancer research program and an associate director for translational research. I'm so honored to bring to the screen Dr. Ben Ho Park.
Thanks so much, Dr. Bailey. Sorry, you'd think by now I'd know that I was muted and had to unmute. This is really a great honor to be talking with you all once again, and I really love this conference. I have a lot of great friends here. And I thought what I'd do is talk to you a little bit about the updates that we've had lately in metastatic breast cancer, and hence kind of the title of my session. I'm going to share screen real quick. In a second. All right. Hopefully you can all see my screen. So really it's the latest news in metastatic breast cancer research. A lot of exciting stuff has come out in the past year, and I thought I'd share some of this with you. Hopefully I'm not going to get into too much details, because I think probably some of the other speakers may in fact have some overlap, and will give you a more granular view of some of the findings or exciting studies that have actually come to maturation in the past year. These are my disclosures. So, when you think about a problem like metastatic breast cancer or metastatic cancer in general, we're often asked the question, why haven't we cured metastatic disease yet? And it really comes down to, first, a lack of knowledge, which is really what research is about, but then second, what we're dealing with. And this has really only been uncovered, I would say, in the last probably 10 to 15 years. We have known all along, and let me try to see if I can do the pointer thing here. We've known all along that, in fact, oops, that's not what I want to do. Cancer is a genetic disease, meaning not necessarily that it's heritable, though we do have forms like that, but that all cancer really at some level is a disease of DNA gone bad. And so when we think about how cancer evolves with time, we often use these types of diagrams or almost trees, where this is a cancer cell, but every time a cancer cell divides, it's not a perfect replica or a copy. You actually get different populations. And this continues to beget even more heterogeneity as these cells divide and diversify and so forth here. So by the time we can actually see cancer, if it's metastasized to a different site, it's already 1 billion cells minimally. And we can throw all the drugs we want on it, but at that point, there's always going to be some sort of drug-resistant population, which will go through what we call a bottleneck or a clonal bottleneck, and then the same process just keeps happening. And in fact, these particular populations are much more stronger, if you will, in terms of the resistance phenotype than things back here. And that's why if we can give chemotherapies up front, like for early stage disease where we don't actually see cancer cells, but we think that they're there based upon historical controls, that's an opportunity to still eradicate what we call microscopic metastatic disease. But for the most part, when we're talking about curing metastatic disease, we're talking about patients who have obvious lesions on their bone scans or their liver and their lungs by CTs or CAT scans. So that's what really makes this a tough problem. There's tons of cells, they're all a little bit different, and we have resistant populations that are going to grow back and continue to get stronger and more diversified. One of the things that we're trying to do in the cancer business in general, and breast cancer specifically, is to be more thoughtful in what we call precision oncology and targeting things that are changes in the proteins, which are really from the changes in DNA. As I said, all cancer is really a disease of DNA. But that's also its Achilles heel, meaning that if you can actually find a drug that works just on the mutant protein or the mutant DNA product, then it won't hurt the other cells. And that's really the thought process for what targeted therapies are all about. It's a little bit more complicated than that, because as I was showing you in the prior slide, you would really want to actually have a drug that is hitting whatever DNA mistake is in here, because that mistake gets propagated. Even though these are all different colors, they all arose from this original cancer cell. And so we tend to call that in our business early driver mutations or truncal mutations, just like this looks like a tree. We want to get a driver mutation where the drug is going to attack this, because in theory, that will be in all the subsequent clones. Now, again, because of the evolving nature of breast cancer and cancer cells in general, eventually you can get resistance in the metastatic setting. But that's something where we've learned a lot about. You can try to chop down the tree at its trunk, so to speak. You can imagine resistance mechanisms, if you use this analogy, would be that these limbs or branches, they may start to swoop down, and that will prevent the axe from getting at the trunk. So that's one way to think about how resistance can still happen, even though we're chopping the trunk of the tree. One of the things that I like to say is we're making progress through research, and I'm going to give some examples. But before I do, it reminds me that I'd like to kind of maybe throw up those questions. I don't know, Dr. Bale, is that possible? I was told just to kind of put them up, and I was going to put up both just for the sake of ease. Do we have? Oh, here we do. All right, so that's not the questions I had. Dr. Bale, is there a separate set of questions that perhaps?
There are. Hold on one second. We're going to get your polling questions up for you.
We can move on until the questions get, here we go. All right, so is metastatic breast cancer curable? And it's telling me the hosts and panels cannot vote, but since I wrote the question, I don't need to necessarily answer. And then I don't know how long this is going to stay on, but there's a second question which will also be relevant in a few slides. Okay, so how are we making progress in metastatic disease? It's metastatic breast cancer, okay, 100% no. All right, interesting. Breast cancer immunotherapy, I think many of you know that immunotherapy is kind of the latest, greatest therapy for a lot of different cancers. And there have been some successes in triple negative breast cancer. Now, we're not seeing the same types of, for the most part, prolonged responses in breast cancer as we do with some other cancers like melanoma or kidney cell cancer. But we for sure have actually had some successes. But in general, breast cancer, particularly estrogen receptor positive breast cancer, is not a tumor type that responds that well to immunotherapy. The other side of that coin is that these drugs can have significant side effects, including death, quite frankly. And so we don't prescribe these to everyone. We want to be very careful who we actually say should get an immunotherapy and who shouldn't. And one of the things that our lab and our centers has been doing for a while now is to actually take patients' tumors and then sequence all the DNA mistakes to try to find who's going to benefit from the right types of therapies. So one of the things that has come out is something that we now know are called tumor mutation burden. And really all that means is that the cancers have a ton of DNA mistakes. Just like I was showing you in those diagrams before, cancers have mistakes in DNA, but some have a lot more than others. And that's what we call tumor mutation burden. And so it can be low, intermediate, or high. We use certain number thresholds to define those, but that's not really that important for this conversation. But what we know now, these immunotherapies, we call them PD-1 or PD-L1 blockades. What they really do, if you look at the pictures here on the right, our immune cells, in this case T cells, have developed mechanisms to distinguish between foreign cells and our own cells. If we didn't have that, we'd all have autoimmunity, and that would not be very good. So part of what we call handshakes are these molecules called PD-1 and PD-L1. And that says, hey, I'm you, don't attack me. And remember, tumor cells are our own cells gone awry. But there's another component where there has to be some sort of protein that looks foreign or foreign enough, and we call that an antigen. And the T cell receptor has to also handshake with that. What these immune therapies have done is they're proteins, they're antibodies, and they just block this particular handshake. And that's enough to make it so that the T cells generally don't attack our own normal cells, although again, sometimes they can, and that's the side effects that I was talking about. But you still have to have a good T cell receptor antigen interaction, something that says, hey, I'm a foreign cell. And so it makes sense. I liken this to playing the lottery. If you buy a lot of lottery tickets, your chances of winning the lottery are much higher. So if you have a ton of DNA mistakes in a cancer cell, and in this case, tumor cell, then there's a higher likelihood that any one of those DNA mistakes will make will make a protein mistakes. Because remember what I was saying earlier, DNA encodes for proteins. And that would be recognized as foreign because that is the Achilles heel, as I said earlier. It's not been ever seen in a normal cell before if it's different enough. And so that allows this kind of differential, if you will, between the T cell and a tumor cell as opposed to a T cell and a cancer cell. So this was put to the test by colleagues at Hopkins, Bert Vogelstein in particular, who was my former mentor, and they had described years ago a particular form of DNA genetic instability, and that was leading to high tumor mutation burden. And sure enough, that's exactly what happened when we looked across cancer types that had this particular type of genetic instability and had high tumor mutation burden. The response rates to these immunotherapies was through the roof, far higher than we'd ever seen before. And that actually put that on the map for immunotherapies and cancers with that particular type of genetic instability. Now, it turns out that particular genetic instability is a method to get to a high tumor mutation burden. But in fact, there are many types of cancers, including breast cancers, that will occasionally have a high tumor mutation burden, but not have that particular genetic instability. And so colleagues across the country started looking at this, including when I was at Johns Hopkins. This is actually a patient of mine who got enrolled on this study. These are, again, patients that didn't have a particular type of genetic instability, but still had a high tumor mutation burden. And this is a patient who had lobular breast cancer, so an estrogen receptor positive breast cancer, and she had had a metastasis in her muscle here, the psoas muscle. And you can see from December 2017 to May 2018, this was completely gone. And she wasn't cured, but she had a disease-free interval of about 18 months where she felt completely fine and there was no evidence of disease. And she used to tell me that, in fact, there was she was at working and nobody could tell that she had cancer, including herself. So that was a really meaningful response. And unfortunately, she did recur. But I think that this highlights that if we really choose our patients wisely based upon the latest information and testing, then we can really afford benefit with minimal toxicity. This has actually now led just 2 months ago to the approval of one of these immunotherapies for adults and children who have a TMB high solid tumor. And so again, my patient was part of that study, which led to this approval. Now, what are some other really exciting findings in metastatic disease this past year? Probably many of you know about this story, but if you don't, and again, one of the talks maybe drive into this a little bit, for HER2 positive metastatic disease, we've actually had a lot of success, and not just in breast cancer, but even other cancers, and in this particular case, gastric cancer. So it turns out that when cancers develop resistance to HER2-directed therapies, many times what happens is that they lose the signaling pathway of what the HER2 molecule does. For those of you who haven't seen diagrams like this before, the HER2 receptor sits outside of the cancer cell. And usually what happens is when we use Herceptin, which is an antibody like this, it blocks this, gets internalized, and shuts down that signaling pathway. And this is the basis of a lot of work from many others and literally decades of clinical research, which led to the approval of Herceptin, Perjeta, and many other drugs now. But once drugs get resistant, many times they still express the protein on the outside of the cell, and it's the inside part, the plumbing, so to speak, that no longer is functioning. And so the cancer cells have learned to actually grow without really using or becoming dependent on the HER2 molecule that they used to be. And so when we say that that happened, we say that they've lost their addiction to that particular gene, in this case HER2. So what this drug does, it's basically Herceptin, but it has about 8 different interesting modified chemotherapies that are linked to it. And so this is very akin to Kadcyla, but this has a lot more molecules. And what happens is that, of chemotherapy that is, when this binds here and gets internalized, all of these chemotherapy molecules get released and they kill the cell. But it also has bystander effects, so it'll even kill cells around it. Now, because of that, it does have more side effects like some of the ones you see with traditional chemotherapies. But this has really been a great new weapon, if you will, in our armamentarium against HER2 metastatic disease, because this is what we call a Trojan horse strategy. It's no longer relying on the fact that HER2 is signaling driving the cancer cell. It's more just of a gateway to get this chemotherapy into the cell and blow it up from the inside out. Now, what about brain mets? So maybe we can flash the other question now. Question 2. Can HER2 positive brain metastasis be treated effectively with drugs? I don't know if you can all see my screen, but maybe that's cheating. Well, I'm going to go on in the interest of time, making sure we have plenty of time for questions. In general, most of the HER2-directed therapies have been antibodies, just like the one I showed you before. Our brains and our whole central nervous system have developed evolutionarily a protective mechanisms to prevent poisons from getting into our brains, because obviously that would be a bad thing. That's what we call the blood-brain barrier. The problem with antibodies is this big, bulky proteins that they do not effectively cross the blood-brain barrier. So even though HER2 disease used to be one of the worst types of breast cancer to have, it's become one of the best because of antibody-based therapies. But what we've seen over the last 15 years or so is that brain metastases have now become kind of the unmet need. Because the antibodies do not effectively cross the blood-brain barrier, it becomes a sanctuary site, if you will, for breast cancer metastases. And that has led to a lot of patients now coming back maybe years later sometimes with brain metastasis who had HER2 positive disease originally. We have had a couple of drugs that are oral drugs that are actually what we call kinase or HER2 kinase inhibitors. Some of you may have heard of neratinib as well as lapatinib, and they may have some activity, but it hasn't ever been adequately studied or proven that those drugs actually effectively can treat patients with brain metastasis. And so that's really been one of the big holy grails that have been sought after in this particular clinical research space. All right, so this again just came out this year from many colleagues across the country, including Vanna Averson, who's a breast oncologist, works with me at Vanderbilt University. And so the drug is tucatinib. This is a pill. It's given both with Herceptin and Xeloda or capecitabine, which is an oral chemotherapy that many of you I'm sure probably know. But if you look at their eligibility criteria, this is really one of the first trials to address directly, hey, let's enroll patients with brain mets because we feel that this has activity to cross the blood-brain barrier. And if you look at these, what we call hazard ratios, everything to the left of this dotted line is a positive, as you see here. Everything to the right is not. And so versus placebo, there's a clear win when it comes to adding tucatinib for treating patients who have either pre-treated with radiation or even active brain metastasis. So this is, again, a huge, huge step forward in our business to really be able to control metastatic disease and perhaps even cure it. So I'd asked this question from the very beginning, and I would concede that the 100% of voters, that's probably mostly the correct answer. But I'm going to give you a little bit of hope and some really exciting things that are happening, not just to this case that I'm going to present to you, but really across the country, and particularly in the space of HER2 positive metastatic disease. There are patients that we have always described for all cancer types that have what we call extreme responses to therapy. And these are patients who have sometimes had complete responses and even get off Herceptin, and they're still okay. And it begs the question, are these patients cured? Probably many of you know the story of Herceptin's development, that one of the first patients who had metastatic disease got on the trial is still alive and well today. And that's really remarkable because we're talking now 30 plus years. And the question always looms though, how do we know these patients are cured or not? And you know, the truthful answer is that we don't. And so my lab and many other labs nowadays across the world have been studying something called cell-free circulating tumor DNA to see whether or not we can actually use this as kind of a window or a lens if patients have residual cancer in their bodies. The concept behind this is actually fairly simple. The blue cells here represent tumor cells, the green cells here represent normal cells, and it's been known for decades that all cells, normal and tumor, shed their DNA as a kind of naked DNA into the bloodstream. We don't know if this has any function. Many people think it's just cellular waste product. People have heard me before calling it cellular poop. But essentially, these cells poop their DNA into the bloodstream and it gets cleared away by the kidneys. And it's depicted here, the cancer DNA, because it's mutated, is all different colors, whereas the green healthy tissue cells liberate normal, uniform green DNA. We can collect this, we can actually extract the DNA out of the blood, and we can actually analyze an assay for it. And this is what we kind of colloquially call liquid biopsies in our business. We started a trial at Johns Hopkins across many different sites nationally where we're looking to see, hey, can we really use this as a measurement of who's cured and who's not? And so these are kind of the future paradigms that we hope to address and really put forward that would allow patients to figure out, hey, maybe after surgery, I don't really need chemotherapy because I'm cured with just the surgery. Or in fact, if I do have microscopic disease lurking about, I should be able to detect it and importantly, see whether or not that level goes down as the chemotherapy is working. If it doesn't work, you would expect it to slowly go up. And that affords us an opportunity to try to give those patients more drugs while they still have microscopic disease that might be curative. And so curing or converting metastatic disease into a chronic treatable condition has really been a goal for many of us in this business. But I want to highlight this case that came across in 2008. This was a young woman who was diagnosed with ER negative, so estrogen receptor negative, HER2 positive breast cancer, and had a lung metastasis that was biopsy confirmed. And this is the CAT scan, or one of her CAT scans, that you can see these two lesions as highlighted by the arrow. And she was given single agent nab-paclitaxel, also known as Abraxane, as well as trastuzumab or Herceptin, and got into a complete remission. Again, here's the CAT scan where there's absolutely nothing. I actually consulted, and at the time, and we still don't know whether or not mastectomy in these patients affords benefit, but we decided to go ahead. In fact, there was absolutely no evidence of tumor in the mastectomy specimen as well. So what we would call a pathologic complete response. And in 2011, basically the patient decided to stop HER2 therapy. We had no data whether that was the right or wrong thing to do. I was admittedly very nervous about it, but in 2017, we finally had technologies where I could actually take that tumor and, even though it was a little bit of tumor, get some DNA out of it. I'm not putting up all the different genes that we found in the patient's original tumor, but one was ERBB2, which is HER2. And sure enough, we found that that was amplified, making it HER2 positive. We also found a gene that our lab helped discover is mutated in a lot of breast cancers called PIK3CA. And this is a very common type of what we call activating or oncogenic mutation in that gene. And in the tumor sample, it was about 30% of that tumor sample, which is expected because usually it's only in about half of the cells or half of the DNA in the cells of the Well, using this technology of liquid biopsy where we can actually take the blood out. We took the blood out in 2017. So remember, now the patient is in complete remission, and using positive controls, negative controls, we dug really, really deep and we did not find a single molecule. Now, can we say that this is equatable to a cure? No, it's one patient. We don't know what this means. That's part of what our study is going to try to address. But this at least affords us the opportunity that if there is disease there, it is below even the level of the threshold of even our most sensitive technologies to date. And that's still the case in 2020, that this is probably the highest level of sensitivity that we have. We're pushing the envelope on that. It may actually change in the next few years. But for right now in 2020, this patient is completely free of disease. And one has to ask the question, is this a cure from metastatic breast cancer? And I will tell you, this is not an isolated story. It is still not a common story, unfortunately, but many of my colleagues have had patients who have had metastatic disease, biopsy proven, get on a therapy and have a response like this. Again, mostly in the HER2 space or HER2 metastatic patients. But there's even a trial ongoing or going to be initiated looking at whether or not there are patients with HER2 metastatic disease that might be curable. And so in conclusions, what I would view metastatic disease in 2020 is vastly different than it was 10 years ago. Patients are living longer with metastatic disease thanks to better therapies developed through research. And a lot of that research has been funded through the generosity of people like you who are doing the money raising, the fundraising, so that it can support the research. That's really what it's all about. We're going to win through research. We still haven't cured most metastatic breast cancer patients. And so again, I concede for the 100% of people who answer that. But I would argue we have probably cured a few. And I would even say more than just a few. Percentage-wise a few, but I think the actual denominator is much larger than that. And I think there's going to be more to come. And that's the really exciting part, is that 10 years from now, I hope to be having this talk and telling you that, we've now crossed the threshold of 50, 60, 70% of metastatic patients are cured. So thank you with that. And I think with that, I also have to acknowledge all the great people in my lab, past and current trainees who did all this work. great collaborators, both at Hopkins and now at Vanderbilt, and especially Susan G. Komen, where I am a Komen Scholar and also receive research funding for a lot of these studies. Thank you.
All right. Well, thank you, Dr. Vinho Park, for such an insightful discussion. We actually have quite a few questions for you. So
Okay.
one question that the Q&A box is lighting up. So one question is what is oligometastasis, and how does this differ from other stage 4 disease?
Yeah, I'm sorry if I mentioned that and didn't define it. Oligo really just means a few. And so when we define patients with oligometastatic disease, first I should step back. There is no universally accepted definition of oligometastatic disease, but I would liken it to maybe less than a handful of sites of metastatic disease, and probably anywhere from 1 to 3 would be a reasonable definition to say that, for example, the patient who might have just a single liver lesion or a single lesion in the rib or something like that is what we would consider oligometastatic disease. We have a notion that the less overall cancer burden, if you will, that perhaps there's still an opportunity to do some good in terms of giving full-blown therapies and maybe in some cases also trying to do curative intent.
Okay, so someone asked a follow-up question. Is it truly considered cured? So now it can be considered chronic versus terminal?
Well, that's a great question. You know, in fact, in some ways it becomes a philosophical one. When we think about, for example, diseases like diabetes, for the most part, we don't cure diabetes, but it becomes controlled so that patients actually live their normal lives and die with the disease rather than from it. That's what a goal of a lot of metastatic disease is right now in the clinic, but I think we'd all like to see the day where we don't even have to worry about that and we can say you are cured. And I don't know if we can do that right now, because the only way currently that you know if you're cured, and this applies to all cancer patients, is if it never comes back, i.e. you die of something else and you never know about it. And that's where the philosophical kind of argument comes in. That's also a big leap of faith. And one of the reasons why we're developing these liquid biopsies, as I showed, it would be really comforting to know that if you had a blood test that said you have a, because nothing's 100% like, but let's say it said you had a 95% chance because we don't detect anything that you're cured, I think that would actually do a lot of good, and it would recognize or allow me to recognize for patients who don't have that, let's say they had a positive result, that those are patients we would follow even more carefully and maybe even offer some additional therapies, either an experimental clinical trial or something else. Okay,
well, thank you. So we have another question. What types of new treatments are available for triple-negative breast cancer?
Yeah, so there's some exciting ones on the horizon, but there's also an exciting one that just got approved. And I think my colleague, who I actually recommended to be a speaker here, may be talking about that, and he was actually a real seminal figure in that trial. But he's probably going to mention a drug called sacituzumab, and there's another name for, or second part of that name. This is also an antibody-based therapy, and it's something that got approved recently this past year, and it's really had some exciting results in triple-negative metastatic breast cancer patients. The other thing that was approved, again, about less than 2 years ago is this immunotherapy that I talked about, atezolizumab, with chemotherapy in triple-negative breast cancer patients who are presenting for the first time. So there are more options out there that are non-chemotherapy. Many of you probably also know there's work done at Memorial Sloan Kettering, Dr. Tiffany Traina, looking at subtypes of triple-negative breast cancer that have androgen receptor expression, and using kind of prostate cancer therapies against the androgen receptor has also been something that is percolating along and also may be a targeted type of therapy for those subsets of triple-negative breast cancer.
Okay, thank you. Another question. I've been living with MTNBC for almost 3 years. I'm on my 3rd line of treatment. How many are out there? And I can't tell if this one is working.
Yeah, so it's a tough question to answer because we approach breast cancer now from the biology of the receptors, and even, again, now even more so with the genetics, like I was explaining earlier. So we're getting more and more refined or granular in how we actually view each individual's breast cancer. That really dictates how many lines of therapy are available. There are numerous chemotherapies that are available for really all the subsets of breast cancer. But as an example, if one has estrogen receptor positive, HER2 negative breast cancer, we have this whole class of what we call CDK4/6 inhibitors, another drug called mTOR inhibitor, PI3 kinase inhibitor, et cetera. So it really depends on the type of breast cancer. And the other thing that's important to remember is that sometimes the receptor subtypes from early stage disease to metastatic can change. And so that's why for most of us, getting a metastatic biopsy at some point is becoming standard of care to recheck those receptors, but then also for me personally to look at the DNA and see if there's anything in that cancer that might be druggable with existing therapies and other types of cancers.
Okay, so one last question. What is on the horizon for ER/PR plus HER2 negative patients? Are there any new research studies for ER/PR positive?
Yep, HER2
Yes.
negative. Yep. So I mentioned, that's the subset I just mentioned about this different CDK4/6 inhibitors, as well as the mTOR inhibitors, and now Palliser, which is the PI3 kinase inhibitor for mutant PIK3CA breast cancers. But there's a lot more. One of the things that we've learned, or really relearned, 2013, is that one of the mechanisms of drug resistance of these cancers to endocrine therapies is that they mutate their estrogen receptor. Remember that trunk of the tree that I was talking about? This is one of those mechanisms where a branch comes out and kind of prevents drugs from working, or the tree from being chopped down. And so there are newer classes of drugs, and interestingly, some are even older drugs that just for whatever reason never made it into the clinics, but may have activity against these mutations, as well as a whole new class of drugs that we're now studying, and a lot of them show promise. So we keep getting to that point where, oh, you develop resistance, we may be able to offer you the next drug and the next drug and the next drug. And interestingly, even amongst the CDK4/6 inhibitors, the drugs that we call palbociclib or Ibrance and abemaciclib or Verzenio, we're actually finding that some patients don't become resistant to all those classes of drugs. So there have been at least 3 studies now published only in abstract form that have shown if you progress on palbociclib and endocrine therapy, you may still respond to abemaciclib. And so this recycling of therapies is also a relatively new concept that we're starting to learn why and who was going to benefit from that.
Okay. Well, thank you. This concludes our session with Dr. Ben Hope-Park.
Thank you.
Thank you again. You've been such a great partner of Komen Chicago. We appreciate you for making time for us and sharing this very, very important information with those who need it. So thank you.